Welcome to Nutrition, Growth and Control of Microbes. This is the first in the series of this, and we're going to be looking at the phases of microbial growth. We're going to be looking at some estimates of microbial growth, so numbers of microbes that have been grown in culture. We're going to look at some of the classifications of microorganisms, and that's based on how well they grow at different temperatures, different pHs, different osmotic pressures, and we're going to look at some of the nutrient requirements as well. And we're going to go a little bit more detail of some of the media that they will grow on. Okay, so it's actually really interesting to look at the phases of microbial growth or to monitor their growth over time. And last week, last prac 1, you looked at culturing and enumerating microbial number. So we're going to look at some of the techniques in a little bit more detail and look, as I said, the requirements for growth. So if you've got a bacteria, for example, that likes to grow at 37, if you put it in the fridge, it may not grow, or it may grow very, very slowly. And that's what happens with some bacteria can actually survive freezing. They might survive. Some might grow very, very, very slowly. But you have to be aware that certain bacteria can grow in quite adverse conditions. So how do microbes actually grow? If we think about a bacterial cell, we've got a very simple cell, remember? So we've got a cell wall, a cell membrane, and then the genetic material. So what happens is the genetic material moves to either end of the cell, and then you get division of that genetic material, and then the cell wall starts to invaginate. And you can see it here coming in and starting to divide that one cell into two. And then after a while, it continues and completes that division. And after that, then you get the production of two daughter cells. Now, as I said probably in one of the first lectures, this can happen really slowly or it can happen really quickly. So things like bacteria like E. coli, in the right conditions, can grow, can undergo binary fission. That's what it's known as. It can do that every 20 minutes. Yes? Okay. And sometimes when they undergo the binary fission, they don't produce two separate daughter cells. If they produce the two daughter cells but they're still joined together, then they keep doing that. They might actually start to form a chain. Or, depending on which plane they're dividing in, they could form a Yersinia or Staphylococcus shape. So it just depends where they, the plane of division. So if you think about how long a bacteria would take to start that division process, that's when you can get an estimation of their growth curve. So you've got 4 phases of a growth curve. You've got a lag phase. So think about a new culture being put, so a culture being put into a new medium, say a broth medium. Okay, it takes a while for it to get acclimatized, equilibrate, and then start to divide. Okay, so we have that lag phase. And then once they all start to divide, they can divide very rapidly. And then you have what's known as a log phase. So that's rapid division. They're using all of the nutrients in that area. They might be producing metabolic wastes, etc. And so that can't, if you think about it growing in a broth, in a flask, the bacteria have got nowhere to move to. Okay, so you get a build-up of toxic waste. They're going to flatten out. Their growth is going to become stationary. And then more build-up of waste, then those cells will start to die. So we have the decline or the death phase. Okay, so that's the type of growth curve that we're expecting. And what do we have? We've got time on the x-axis and the number of cells, the log of the number of cells on the y-axis. Okay, so just please try and keep a picture of this in your mind. Now, if we go through the lag phase, so this is a recovery phase. So it's lagging just before that exponential growth. And the cells are becoming active metabolically, and they're just getting ready to start dividing. And you can get upregulation of particular genes in the bacteria as well. Sorry, microorganisms. And so if you get that upregulation, you can get the essential nutrients being, more essential nutrients being generated, so you're getting the cells ready for that division. As I said, in the log phase or the exponential growth phase, you get cells in the optimal growth state. So they're nice and metabolically active, and they repeatedly divide via binary fission. And if you're thinking about doubling time, it can vary from about 20 minutes, like E. coli in a perfect situation. It may take several days. So some microorganisms are very slow growers. Okay, so if you think about when we incubate a plate, we might incubate it for 24 hours, or it might take up to 5 days, depending on what we're selecting for, what particular organism we're looking for. So you probably don't know yet, you possibly do. Everyone's heard of stomach ulcers, yes? They are caused by a bacteria known as whose name is Helicobacter pylori. Okay? It's very hard to grow, and it's a very slow grower. So the people in WA who first described it, they had a culture of mucosal lining of the gut, and they were growing it. And then they forgot about it. It was a long weekend or something like that. And they, we'll have a look at it when we get back from holidays. It was only after that elongated incubation that they found something growing. Okay, so some can be very slow growers. And some are very hard to culture naturally, like in the laboratory. So they need special types of media. So in the stationary phase, we've got the number of new cells equaling the number of dead cells. So there's no continuation of that log phase. And it may be, as I said, if the bacteria, the microbes have gotten, they've taken up all the nutrients or most of the nutrients, and they're accumulating those waste products. And remember, if there's sugar, some sort of carbohydrate in that agar, they may be fermenting it and building up acid in the environment, which is quite detrimental to growth. And in the death phase, as I said, this is when the cells start to die. They can't actually regenerate, undergo binary fission, and therefore you get a decrease in the viable cells. You might still have cells there, but they're not able to reproduce at all. They're just dead. Okay, so viable cells is what we're looking for. And if you wanted to store a bacteria, for instance, so maybe you've got a culture that you want to keep for a year, 10 years, there's a number of different ways you can keep that. It might be that you incubate or produce a culture in log phase before it gets to that stationary phase, and then put it in the fridge. Okay? Sometimes you can get the cells and freeze dry them, and then you can store them for a long time. And then when you are ready to use them again, you put in nutrient broth typically, and grow them up gradually like that. Okay, so there's a number of ways of keeping bacteria. So, but when we start to grow them, sometimes we just need to know how many are viable. So how many are living? And there's one way we can do that is a viable cell count, but sometimes you might just want to look at a total cell count. So how many cells were actually there? They might not be viable, but they're still there. And what you can do is put, we'll just put that there. So there's particular slides that you can put under the microscope, okay, that have grids in them. Yeah, so you put a known volume of culture in broth onto that little indent or that little well, put a cover slip over it, and then you can count them under a microscope. Okay, so that is just known as a total cell count. And that cytometer is known as is the Petrov-Hauser cytometer or hemocytometer. So you can understand a hemocytometer. These are also useful if you're staining blood cells, and then you can count the number of white cells or the different types of white cells. And you know with those hemocytometers, it's a certain volume in that well. So you can work out how many bacteria or how many cells are in that particular sample and therefore in that particular volume. But if we want to know how many can grow, we need a viable cell count. We need to grow them on a particular media to see how many cells will grow. And usually you use what's known as a standard plate count method. So standard, it's just a basic nutrient agar. Okay, so it's the standard conditions. And what you're looking for is the formation of colony-forming units. So that colony-forming unit, and you'll see those in your next prac, is an area where there's a lot of cells in one area, okay, together. And that colony-forming unit might arise from one cell, or it might arise from a group of two or more cells, okay? But standardized terminology is a colony-forming unit. And you can get the number of colony-forming units in a broth or a sample of soil or anything like that by a number of different methods. You can use a spread plate method or a pour plate method, and you've done both of those, or you will do it in Prac 1. When you let those set, then you incubate them for a standard amount of time in a standard environment. So it's usually 37 degrees for 24 to 48 hours. And when you get the plates out of the incubator, you only, with the pour plate method, you only count the plates, that dilution, with between 30 and 300 colonies. Okay, that range gives you the best estimation of the viable cell count. But So with the spread plate method, you typically count only those plates with 10 to 100 colonies or CFUs per plate. But it takes a lot longer because you need incubation. Now I'm talking about in this course and in the pracs, we're going to be using classic methods. Okay, we're not going to go into the genetic methods that can be used. If you continue on with MedMicro, you'll be looking at far more detailed and current methodology. But it's important for you guys to know the basics, so using the classic ways of doing it. And everyone should be well and truly familiar with the serial dilution, yes? Because what we're doing is we have a sample and then we're diluting it in series so we get a dilution where we can get countable plates. Okay, that's the process behind that. And then once we get the dilutions, we can use those dilutions to make a spread plate or a pour plate. So, and I'm sorry about that, I didn't draw this. So a spread plate method, you put a certain volume on a plate, typically via a pipette, not just tipping, and you can pour, say, 0.1 of a mil on there, and then you spread it around. But for this you need an agar plate that is solidified. So you're just spreading it on the agar, and when it grows, it's just at the top there, okay? Then with the pour plate method, you can put some solution, so some from your serial dilution, and then you pour agar onto it, you mix it, and then the colonies will be throughout the agar. And obviously, if you've got a dark agar, you might need illumination to count them. Okay, so you might, from if you did triplicate, you might have plates where you had 78 colonies on one, 83, 81. So they're countable, and so you would count those and take an average. Okay. Now, what's with the spread plate? You have to spread the bacteria over the surface of the plate somehow. And so what you can do is use glass or metal rods, so known as hockey sticks. And obviously if they're sterilised, that's great, but if you need to, you can flame sterilise them. Typically, if you have plastic spreaders, they already come pre-sterilised, so you don't have to flame them, which is a good thing. And you wouldn't want to flame plastic anyway. And this is a really good method to use because you can, if you are experienced in doing it, you will get separate colonies. However, sometimes it does take time because you've got to bake the plate, dry the plate, and then spread the plate. And if you're not used to doing spread plates, because it's quite a technique, isn't it? You might not get the proper separation. Now, sometimes you can have highly motile bacteria on there as well. So if you get a little bit of condensation on there, so it hasn't dried into the agar, if you've got a motile bacteria, it might swarm across the complete surface of the plate. And that's known, and if that happens, then there's no way you can actually have a proper count. So there's a number of disadvantages. With the pour plate, you typically get well isolated colonies because you've mixed them really well before the agar sets. With the colony size, they might vary. So you've got the colonies being formed throughout the agar. Even though it's not very thick agar, sometimes the oxygen might not be enough for colonies to form, or the heat from the agar might actually kill off some of the bacteria that are susceptible. Okay, so there's a number of different problems there. But if hopefully we've got a good dilution, we've incubated the plates at 37 degrees overnight, so 24 hours, and you're counting a plate between 30 and 300 colonies. And you can see that some are less easy to count and some are more easy to count. Okay? And to count them, sometimes you've got a plate counter, which basically you put on over a graticule, and so this grid behind it, and then you've got light illuminating it. Okay, and obviously you can see. You certainly wouldn't count this plate, but you can see that there's different types of bacteria there as well. So the large red ones, the smaller white ones, there's tiny, tiny little ones as well. So that's probably well over 300. But saying it, when you counted them, you had about 175 colony forming units. Okay? But that was from the plates from the 10 to the minus 5 dilution. Okay? So how do you work out, how do you go back to how many there were per mil of the original? You have 175 by 10 to the 5 CFU per mil. Everyone's happy with that? And then you want to get that in scientific notation. So you have 1.75 by 10 to the 7 CFU per mil. Everyone happy with that? Yes? Well, you'll be getting some practice, I think. Now another way, so that's actually counting the colonies and knowing that there are caveats because not all bacteria are going to grow. Some bacteria are going to grow more slowly. Some bacteria might be affected by the temperature of the agar, etc. So there's a number of different caveats. So one other way of estimating growth is you can look at the turbidity of a broth. And so you can, look, as they grow, as a cell grows in a broth, then from the broth is kind of clear to start with, and as they grow, it will become more and more turbid, okay, because of the light scattering properties of the cell membrane, the cell wall. And typically, if you just swirl a culture gently, you can sometimes see the swirl of cells coming up. And if you've got a cloudy, if it's slightly turbid, it's about 10 to the 6 colonies or cells per mil, okay? But more accurately, you have to deal with cultures over 10 to the 7, okay? And this is what you get. Now, it's just guesstimating, okay, looking at the turbidity. So on the right-hand side, you can see the lines through that broth quite easily. So it's quite clear. So you would have no turbidity. So there might be cells there, but it's estimated to be less than or equal to 10 to the 7 cells per mil. Now, on the left-hand side, it's quite turbid. And so, depending on the level of turbidity, and there are scales, the Macfarlane scales of turbidity, you can guesstimate what the concentration of cells are. Okay, obviously a disadvantage of this is it's very subjective. Okay, rather than a count, you've got quite a subjective thing. But There's another way you can check on the turbidity, and that's measuring the absorbance of the broth. So you can actually get an absorbance for each broth. Sometimes it might be absorbance of broth over time, so you can construct a growth curve. And for this, you need to use a spectrophotometer, and depending on the wavelength depending on the broth, you need different wavelengths. And it's a really good measurement of cell number or cell growth. And it's quick and it's relatively easy. So quick to put in a spectrophotometer, it's quite easy. Now, there are other factors that affect bacterial growth, like temperature. So everyone has probably grown something in their fridge, an experiment in the fridge. Yeah, so temperature does affect growth. Oxygen does affect growth as well. So you've probably heard the terms aerobe and anaerobe, or facultative anaerobe. That means it can do either. pH, some bacteria will grow in low pH, some don't like low pH. Osmotic pressure, some can grow in high salt or high sugar, some can't. So there's a number of different factors that can affect the growth. But we're going to start off with temperature. And each particular species of bacteria has their own preferred temperature range for growth. And why is that? Because the enzymes inside the cell actually will work maximally at that range, in that range. And so we can look at it at three main reasons, so that there's three critical temperatures. There's the optimum temperature. That's where you have the fastest growth occurring. So all the enzymes are working optimally and you get the optimal binary fission. The minimum temperature is the bottom. So this is where growth only just occurs, and below that, no growth occurs. The opposite is maximum temperature. So the cells might be growing, but very slowly, but above that temperature, there will be no growth. Okay, so they're the three critical temperatures. And you always have to think about the enzyme activity. So that's what's driving some of this. And enzyme, remember, is a protein. So at high temperatures, that protein, which is beautiful and globular, can just fall apart. And if it falls apart, then it can't have any action. And so we know those three, the optimal temperature, the minimal and the maximal temperature, but we can classify the bacteria in those growth ranges, or those temperature ranges. So we have the psychrophiles, where they grow 15 to 20 degrees. They can grow a lot lower as well. The mesophiles, their preferred range is 25 to 40 degrees, and you can see that the human pathogens fall into that category, so their optimal temperature is 37 degrees. Thermophiles, these are the ones that can grow at 55 to 60. Sometimes they can survive 80 or more, or boiling. So the thermophiles are really okay with higher temperatures. Some of the extremophiles, so the extreme thermophiles, can survive 115 degrees. So the archaea in springs, hot vents, and hot springs. Okay, so we've got quite a range. And if we go the other way, the psychrophiles, some have been found, some yeasts have been found in Arctic ice as well. So we've got quite a range, temperature range to deal with. And if we look at some typical ones, so typical psychrophile, you can see the growth range here, and the optimal temperature is about 15. So Flavobacterium is a typical psychrophile. Then we've got a typical mesophile, and you can see that growth curve or range is quite extensive. So things like species like Escherichia are a typical mesophile. Then we have a typical thermophile and an extreme thermophile, and it can go up even further. Okay, but they're the classifications that you need to be aware of. And so the psychrophiles, these are ones where you have the enzymes and the ribosomes, so producing the proteins, they're active at low temperatures. And they can actually be, if the temperature goes up above about 25 degrees, it might stop those enzymes working completely. And there's a lot more lipids and unsaturated fatty acids in the membrane, so it keeps the membranes semi-fluid. So you don't want them to be freezing, okay? So you need that fluidity. The thermophiles, these are the enzymes, they are quite stable at that higher temperature, and the ribosomes producing the proteins are quite stable at that higher temperature as well. And they have quite a neat trick with their proteins. They have a lot of what's known as the salt bridges in the proteins. So these are basically, they increase the thermal stability of the protein. So remember I said proteins can fall apart, but the salt bridges actually hold them together a little bit more. So they have that increased thermal stability. And in the membranes, they have a lot of long-chain fatty acids and saturated fatty acids. And so they work really well at high temperatures. But if we took them out of the high temperature into a lower temperature, then they would basically solidify the membranes. Okay, so they're adapted to that high temperature. Now the oxygen, we've got some organisms, some of the microorganisms they need oxygen. They're obligate aerobes, so they can only grow when there's oxygen there. The facultative anaerobes, these are the ones that can grow with or without oxygen, okay? But they grow better with oxygen. Then we've got another classification. We've got the aerotolerant anaerobes. These are the, they can survive with some oxygen, but they metabolize their substances inside the cell anaerobically. So these are the aerotolerant. They can put up with a little bit of oxygen, but they choose to metabolize anaerobically. Then there's the obligate anaerobes. Obligate, if there's any oxygen in that environment, it will kill them. Okay? So obligate anaerobes are the ones that die in the presence of oxygen. And we've got another classification, and I probably should rearrange the order of these, but we've got the microaerophilic. That means that they only need a small amount of oxygen, so concentration of about 2 to 10% to be able to grow. And if you think about normal atmospheric oxygen, what we're breathing now, we've only got about 20% oxygen in the air we breathe. So the microaerophils are quite well suited to a low oxygen environment. And part of that is they could be capnophiles as well. These are the ones that love carbon dioxide. So the phials are loving. So there's a number of different definitions, so different divisions of oxygen requirements. And if we looked at that in growth pattern in an agar or a broth. If we just used a liquid broth, okay, and didn't disturb it, we didn't shake it violently or swirl it gently, we just left them growing as they are, we would have bacterial growth. If it's an obligate aerobe, it needs oxygen. So it will always be up the top. The growth will be close to that, well, it will be at that junction of the broth and the air. An obligate anaerobe will be growing as far away from the oxygen as possible. And so you would typically find growth at the bottom of the tube. A microaerophile can put up with a little bit of oxygen, so it would be growing just a little bit further down, away from the oxygen, but not right down the bottom. And a facultative anaerobe, it will be growing everywhere because it can grow anaerobically, it can grow with a little bit of oxygen, and it can grow with a lot of oxygen. So just looking at the growth profile in a tube, you can, with no disturbance, you can actually tell what their growth requirements are, oxygen requirements are. But why do we need anaerobes, obligate anaerobes? Why would they be beneficial? So they with oxygen, remember I said oxygen can be toxic? So if you're a bacteria and you grow without oxygen, then you don't have those toxic oxygen radicals, so you don't have to produce catalase, etc. And you don't have to use any of these, okay, in your metabolism. So aerobes, so the ones that need oxygen, they have to have the enzymes to get rid of those toxic oxygen. So the two enzymes that they use are the superoxide dismutase and catalase. And catalase we looked at before. So if we've got one of the oxygen radicals and hydrogen, and we've got this enzyme, that will produce oxygen, but it will produce hydrogen peroxide as well. And that is not good for cells. So what happens is that hydrogen peroxide is acted on by this enzyme catalase to produce oxygen and water. Okay, so it's gotten rid of the oxygen radicals, the toxic oxygen. It's produced hydrogen peroxide, and then they get rid of the hydrogen peroxide just by a two-stage thing. And if you had E. coli, okay, which is a facultative anaerobe, if it's mutated so it loses these two enzymes, what's going to happen? It's going to behave like an obligate anaerobe. It's just going to stay away from the oxygen because it doesn't, or if it goes near the oxygen, it doesn't have those enzymes to get rid of it, the toxic oxygen, and therefore, if it doesn't have the enzymes, it will die. But if it behaves like like an obligate anaerobe, then it can survive. Do you see that? Make sense or not? Okay, silence. Okay, so sometimes we want to culture bacteria in an anaerobic environment. So get rid of all the oxygen and see what can grow. Or we want to make sure that we give as much oxygen as possible. So we can culture either aerobically or anaerobically. So aerobes, if you're culturing aerobes, you can gently shake the culture broth or whatever, so you get a lot of oxygenation of the broth. Sometimes you can even bubble air through, like sterile air through, to make sure that there's more oxygen there. Or if we wanted to get rid of the oxygen, we have the capability of putting the plates in jars and getting rid of the oxygen in the jars. So it might be that we burn a candle, so it burns off all the oxygen and then you're left with no oxygen. Or these anaerobic jars where you have some chemical packs and they will chemically get rid of the oxygen. They're like oxygen scrubbers. So there, we will be using these types, the gas pack ones, anaerobic jars, in the pracs. Now, pH is another thing that you have to worry about with bacteria. So you've got bacteria that love acid environments, ones that love neutral environments, alkaline environments. Sometimes the extreme alkaphiles might be found at pHs 10 and above. Okay, so if you think about the pH scale here, we've got the extreme acidophiles, acidophiles, neutrophiles, alkaphiles, and extreme alkaphiles. Okay, so typo, Kate, that's bad. So you need to be aware that they can grow at different pHs. And that's important because if you are selecting for a particular type of organism, you might want to look at the pH of the agar. So you're selecting the ones that will grow at that pH. And what happens with pH, it can affect growth quite a lot. So if you consider most bacteria, they will mostly grow at a neutral pH, okay? But some have at the extreme ends. Now, that's bacteria, but fungi, they're a different class, a different case, because they prefer it slightly acidic. And when we're looking at we're trying to grow particular types of fungi, we could use something called malt extract agar, and to acidify it, they're acidified with some lactic acid. So you put some lactic acid in there as well. Okay, so you've created a pH environment that fungi would like to grow in. Now there's another one you could use, a different type of agar known as dichlorin rose bengal chloramphenicol agar. Now it's a bit of a mouthful, but chloramphenicol is basically antibacterial. Okay, so you are selecting for fungi and trying to get rid of any bacteria. So you've got an antibiotic in there, basically. The pH is not as low as the malt extract agar with lactic acid, but that's probably why you need that chloramphenicol as well. So you're discouraging any sort of bacterial growth. If you do that, then you've got fungi that can grow there without the competition from bacteria. That's all I was going to say. And then there's sabrodextrose agar. pH about the same, and this is one of the agars we used in Prac 1. A lot of fungal media have antibiotics in them. Okay, why? Because if you don't have antibiotics and you've got a specimen, you're testing a solution or some sort of food stuff or whatever, and you grow it for, say, 24 hours. You get a lot of bacteria growing, but the fungi take a lot longer to grow. So if you've got a lot of bacteria growing first off, they're taking up all the nutrients producing wastes which may actually be detrimental for the growth of the fungi. So that's why you try and get rid of the competition before you plate out the sample. Okay? Osmotic pressure is used as well. So remember the what I spoke to you about salt concentration? So that's one of the osmotic selection criteria that we can use. So most cells require a certain amount of free water to grow. If you can change that free water that's available, then you can be selective for particular types of bacteria. So if you've got just a normal ordinary bacteria that likes isosmotic conditions, so normal osmotic conditions, if you then put it in a hypertonic solution or environment, then the cells would shrink and stop growing. So why? Because if it's a hypertonic solution, the water inside the cell is going to move out into the environment. So the cells are going to shrink and they can't grow. If that cell is put into a hypotonic environment, where's the water going to go? It's going to move into the cell is going to swell and it's going to lyse. So you can see this phenomenon with human red blood cells as well. They react the same way. So some bacteria can grow, survive and grow in hypertonic or hypertonic solutions or environments because they produce particular things inside the cell that can balance the osmotic pressure. Yes? And that's exactly what staph does. So Staphylococcus species grows on skin where salts are quite common. And it can tolerate up to 10% salt in solution. So the culture media, not many other bacteria can tolerate that, okay? So the culture media typically has about 7.5% salt, and to differentiate between Staph aureus, which will grow, and Staph epidermidis, which may or may not grow in that environment, there's different indicators there that will cause a colour change with the metabolism of the bacteria. And so some bacteria require very high osmotic strengths for growth. So the halophiles, these are the salt lovers who grow in salt lakes, etc. And if some of them, how many people have done chemistry? Have you got an idea of how much 3 molar sodium chloride solution is? How concentrated that is? Some of these halophiles actually won't grow if the salt concentration is less than 3 molar. Yeah? So that's why they go into the extremophiles. We also need, we've got the different environments, but we also need lots of different nutrients for the bacteria or the microorganisms to grow. So the most important ones are carbon, nitrogen, oxygen, hydrogen and sulfur. There's some ions as well that they need. So most microbes are the chemoheterotrophs. And the bacteria and fungi can use those large molecules as nutrients and produce enzymes that break them down. Now in this case, they produce exoenzymes, which means that the enzymes go out of the cell itself and break down those complex molecules outside the cell, and then they can get those molecules, the smaller building blocks, and absorb them into the cell itself, so they get those nutrients. Okay? If you think about, you know, that beta hemolytic bacteria that I showed you before that was breaking down the red cells, yeah? That's basically an exoenzyme. So it's being produced in the cell but put out of the cell to have an effect. And then the breakdown products are taken back into the cell for use. And hopefully you'll get an understanding that pure cultures are very important, because bacteria, if you've got a mix of bacteria, they might all be wanting different things and producing different things. Yeah? So that's why if you're going to identify a bacteria, then you need to have a pure culture. Now, once you have a pure culture, then you can look at that species specifically. And what you can do is, if it's a gram-negative Enterobacteriaceae, you can use what's known as an API strip. Okay, so all of those single tubes that we looked at, this is all in one strip. So you've got different tests here. We've got H2S. So here we've got before it's negative, and here we've got a positive, so it's quite black. So what happens is you get these strips. They come like this, basically, and then you have a pure culture. You grow up a pure culture and put a little bit into each of these wells. They're like little cups, and then so a cup here, and then they go down into a bit of a well, and they're filled with reagents. So you put it in here, and it gets taken down into that, or mixes with these reagents, the liquid in the things, and then you incubate it for 18 to 24 hours at 37, and then you read the reactions. So if there's no reaction, then it's kind of clear, or there's specific colour changes that occur. So if we look at back H2S, you've got no production of H2S here, but here it's gone all black. So that's a positive reaction. If you've got urease, it's a negative here, so it stayed yellow. But here we've got the production of urease, and so it's gone pink. So the colour reaction tells us a lot about what's happening. So here it says gel down the bottom. You probably can't see it here, but it says gel, and that just means that there's gelatin in there. And it doesn't break down gelatin here, so it doesn't produce gelatinase, but here it does, and with that breakdown product it produces a black coloration. Here, these are the sugars. Okay, so it's quite a range of sugars, and you can see that there's been no metabolism occurring, so they stay the same colour. But here we've got metabolism occurring, and they've changed they've produced acid and changed the indicator. So it's like, oh, well, that's very pretty, so what do you do with it? So you have each of these in triples. In threes, you have numbers associated, whether it's positive or negative. And then the outcome is a particular number, and you look it up in a table, and it tells you 98% positive for 98% chance that it's a particular bacteria, or it gives you the percentages of how accurate it might be. So that's a really nice test to use, but you need to get that pure culture. So with the pure culture, you can use a streak plate, and so you get individual colonies here, and when you have an individual colony, you might grow that up and put that on your API strip or any other sort of biochemical strip you might be using. If you're using a pour plate method, sometimes it might be very difficult to do that, so it's much easier to get a pure culture on the top of an agar rather than embedded in an agar. And the culture media itself can be either synthetic, so you know exactly what's in there. It can be quite complex, or it could be quite simple. So nutrient agar is one of those relatively simple workhorse type agars. So you get growth occurring. But sometimes you need to put in serum or blood or something like that to select for particular organisms. And that's what's known as the diagnostic media. So once again, you've got selective, differential, and what we didn't go through before was enrichment. So, and that's beautiful because the different organisms are written there in bacteria, and you can see the different colours. That's what they look like on that particular type of plate. Okay, so the enrichment basically takes a particular substance that the organism needs, and so that organism will grow preferentially. Okay, it's selected, it's enriched for that particular organism. So things like blood agar, so normal agar with a particular, a certain percentage of blood, or chocolate agar. No, it's not what you think it is. It's actually blood agar that's been heated. Okay, chocolate agar is one of the types of agars that you grow Neisseria on. Okay, so you have particular growth types on chocolate agar. Or, and so this is chocolate agar overgrowth. So it's a rectal specimen, and just putting it on chocolate agar might cause overgrowth. But if you had chocolate agar with certain additives known as Thayer-Martin agar, then it will only grow Neisseria. Okay, so you can select and then select in an enriched environment. And this is, you'll find some of the different colours in your appendix as well, but this is quite a nice way to look at the diagnostics. So you've got brilliant green agar, which is one of the agars you use for salmonella. You've got EMB, eosinomethyl blue, and you'll be able to see your streak plates from PRAC1 using EMB. And E. coli is really, really beautiful on that because it produces a metallic green colour on that EMB. And MacConkey agar that we also used, and that's for gram-negative enterics. Okay, so the bacteria that typically grow in the gut. And as I said, this is TSI, triple sugar iron, and when you look at the particular colours, so you've got what's happening in the slant, in the butt, if there's gas and if there's H2S. Yeah? Just from this one type of agar, if we looked at this one, it's the one at the end, you've got a red slant, okay, so that means it's alkaline, and it's got a yellow butt. You can't really see it because it's all H2S, so it's a yellow butt, so it's acid. Then have you got gas? Yes, there's gas, so it's positive for gas, and it's positive for H2S because it's really black. It's most likely Salmonella or Proteus. Just looking at how their biochemical profile, how they will metabolize certain sugars, and the iron in there as well. So we've looked at the growth phases, and you understand the different phases of growth, the growth curve, and you've got how the numbers of microbes can be enumerated as well. We've looked at the different types of environments that they need to grow optimally and what they can survive as well, and how you can use their metabolism to identify them. Okay, so has everyone got any questions about that? Yes? Yeah, the thermophiles, the extreme thermophiles.
You mentioned that the brain solidifies at like one temperature.
Yes.
Temperature, does it like become semi-solid?
No, so the question was, with the extreme thermophiles, their cell membrane actually becomes solidified at normal temperatures, or what we would consider normal temperatures, but at the temperature that they're normally growing at, it's quite fluid. So if the temperature decreases a little bit, then the membrane is not so fluid. Yep. So before you go, practice exam questions. Okay. Everyone got an answer for 1 and 2? Yes? Are you right? Excellent. Well, I will see you next time. Thank you.